I now call to order the Society's 2,448th meeting in the 150th year since its founding on March 13, 1871. Good evening, everyone. My name is Larry Milstein. I am the president of PSW, one of the oldest scientific societies of Washington, D.C., committed to providing a forum to further scientific understanding and inquiry. Welcome to our members, guests, and friends to tonight's lecture by Romy Amaro. The Society has been meeting and hosting lectures via Zoom for over a year, starting with the Joseph Henry Lecture on May 15, 2020, almost a year and a half ago. Meetings this fall thus far have been done via Zoom as well, due to COVID-related restrictions here in DC, and also restrictions on speaker travel. But that is coming to an end. Our next meeting on Friday, November 19th, will be in person with the speaker in the John Wesley Powell Auditorium of the Cosmos Club in Washington, D.C. Subsequent meetings will be held in person when possible and otherwise by Zoom. In-person meetings will adhere to the COVID-related rules of the District of Columbia and the Cosmos Club. Proof of vaccination, including a picture ID, will be required for in-person attendance at the November 19th meeting. The Cosmos Club rules do not provide any exceptions. The meeting will be live streamed to the PSW Science YouTube channel as always, and YouTube viewers will be able to ask questions in the YouTube chat box. Additional details will be posted to the PSW Science website and will be sent to PSW members via email. Everyone, please check the website often for up-to-date information. The Society is grateful to the sponsors of the 2021-2022 lecture series for their support. The Policy Studies Organization, in cooperation with the American Public University, and a generous donor who has asked to remain anonymous. We thank them all. Thank you. I am pleased to announce the following new member, Romy Amaro, tonight's speaker, whose interests will be clear to you in part from tonight's proceedings. We welcome her to membership. If you are not a member and would like to support the Society, you can do so through the PSW website. And if you would like to join, you can do that through the PSW website as well, using the blue join button and following the prompts. We invite you to join and we welcome new members. Member at Large Connor Nixon, standing in for Recording Secretary James Heelan, who could not be here tonight, will now read the minutes of the 2,447th meeting and the lecture by Fengshuan Liu on the 30-meter telescope, 12 times Hubble and halfway to space. Connor, the screen is yours.
Thank you, Larry. On October 22nd, 2021, by Zoom webinar broadcast on the PSW Science YouTube channel, President Larry Milstein called the 2,447th meeting of the Society to order at 8:02 p.m. He welcomed everyone, thanked sponsors, and announced new members. Recording Secretary James Heelan then read the minutes of the previous meeting and the lecture by Olivier Guyon on adaptive optics. After soliciting comments, announcing that the minutes would be posted to the website, and thanking the Secretary, President Milstein introduced the speaker for the evening, Fengshuan Liu, Project Manager of the 30-meter telescope project. His lecture was titled 12 times Hubble and Halfway to Space. Liu began by discussing how telescopes have progressed in size over the last 400 years, from the earliest instruments just a few centimeters in diameter to today's largest 10-meter class telescopes. He noted that each increase in size and light gathering power have enabled new discoveries about increasingly distant objects. He said that the next generation of 30-meter class telescopes promise to bring about dramatic new discoveries as well. The speaker began by discussing the history of telescope building. In particular, he noted that the building of the Hale telescope demonstrated that there are insurmountable problems of casting and polishing single monolithic mirror telescopes beyond a certain size, and that the maximum size was being reached by the Hales mirror. While it has been possible to cast up to 8-meter class telescopes, mirrors much larger than that cannot practically be made by modern fabrication methods. Fortunately, Liu explained, the limitations can be overcome by segmented mirror telescopes, in which multiple, relatively small mirrors are so perfectly aligned that they act together in effect as a single large mirror. The idea was conceived by Jerry Nelson and first demonstrated in the Keck telescope, which used laser sensors and computer-controlled actuators to align 36 1.8-meter hexagonal mirrors, forming, in effect, a 10-meter aperture primary mirror. The Keck telescope paved the way for the current generation of extremely large telescopes, now on the drawing boards and under construction. The speaker emphasized why telescope size is important, both for enabling us to see more distant, dimmer objects, but also for resolving smaller angular scales. To illustrate this, he pointed out that a telescope with a 30-meter primary mirror can see objects 12 times smaller than a telescope with a 2.4-meter primary mirror, which is the size of the mirror in the Hubble Space Telescope. However, Liu told the audience size is not the whole story. The resolving power of telescopes on the ground are limited by distortions introduced by the Earth's atmosphere, something that does not limit the performance of space telescopes, and is the reason observations by the Hubble Space Telescope outperform Earth-based observatories, despite its relatively small mirror. Atmospheric distortion can be limited to some extent by choosing the right location, he said. For instance, the peak of Mauna Kea on the Big Island in Hawaii is halfway to space. Only half of the Earth's atmosphere remains above the summit, dramatically decreasing the interference. Although high altitude helps considerably, it does not entirely eliminate atmospheric distortion. Liu explained that much of the distortion can be corrected by adaptive optics, a technique developed around 30 years ago that has revolutionized astronomy. He described the principles behind adaptive optics, in which the shape of a deformable mirror is adjusted in real time, so as to just compensate for distortions introduced by the lights passing through the atmosphere. The distortions are detected using a guide star. Both natural stars and artificial point sources created by lasers in the upper atmosphere can be used. The speaker said that the 30-meter telescope is designed to answer the big questions, including the nature and composition of the universe, how galaxies and black holes form, and which came first, the nature of exoplanets, and to assist with the search for life in the universe. Liu showed examples of simulated observations with a 30-meter telescope, illustrating how it will dramatically improve our view of solar system objects, exoplanets, stellar motions, distant galaxies, gravitational lensing, and the first stars and galaxies. Liu then turned his attention to the technologies and building of the telescope. 82% of the key subsystems are already designed, he said, including the dome, which will be provided by Canada, and is designed to minimize its impact and footprint on Mauna Kea. The telescope is comprised of 492 mirror segments covering 652 square meters, with gaps between segments of only 2.5 millimeters. The polished mirror segments are provided by the US, Japan, India, and China from blanks prepared in Japan. Liu said that every 2 years, the mirror segments will need to be recoded. 10 segments will be replaced every 2 weeks during daytime when the telescope is not being used by a specially designed robot. In conclusion, he said that the TMT design is mature and well understood and promises to bring new discoveries and to change the landscape of astronomy for the next 50 years in the Northern Hemisphere. The speaker then answered questions from the online viewing audience. PSW member Carl Merrill asked the speaker to contrast the capabilities of the 30-meter telescopes to the James Webb Space Telescope soon to be launched. Liu responded that Webb is a space telescope operating in the infrared part of the spectrum at longer wavelengths, where it is necessary to go to space to avoid the emission from the Earth's atmosphere. TMT, on the other hand, is an optical and near-infrared telescope that can cover those short wavelengths from the Earth. Member Dave Rabinowitz asked whether the altitude of the guide star needs to be modified based on atmospheric conditions, and if so, how rapidly. The speaker responded that the guide star is located at around 90 kilometers and is not a specific point, but in fact a column. The frequency of the adaptive optic system modulation is 1,000 hertz, which is suitable for the CMOS camera, which operates at 600 hertz. After the question and answer period, President Milstein thanked the speaker, made the usual housekeeping announcements, and invited guests to join the society. President Milstein adjourned the meeting at 9:32 p.m. Temperature in Washington, D.C., 17 degrees Celsius. Weather, cloudy. Concurrent viewers of the Zoom and YouTube live stream, 86. And views on the PSW Science YouTube and Vimeo channels, 480. Respectfully submitted on behalf of Recording Secretary James Heelan by Connor Nixon, Member at Large.
Thank you, Connor. The minutes will be posted to the website in due course. Please send any corrections or comments on the minutes to corresponding secretary Robin Taylor at correspondingsec@pswscience.org. We now turn to tonight's lecture, Computational Microscopy of SARS-CoV-2, using simulations to see the unseen. It's my pleasure to introduce tonight's speaker, Romy Amaro. Romy is professor and Romy is distinguished professor and endowed chair in the Department of Chemistry and Biochemistry at the University of California, San Diego. Previously, she was a member of the faculty of the Departments of Pharmaceutical Sciences, Computer Science, and Chemistry at UC Irvine. Romy's research is broadly concerned with the development and application of state-of-the-art computational methods to questions in drug discovery and molecular-level biophysics. In particular, she is keenly interested in developing new multi-scale simulation methods and novel modeling paradigms that scale from the level of atoms to the level of whole cells. Recently, she has been applying these methods to the SARS-CoV-2 virus to understand how it binds to and enters cells, with special attention to the spike protein. And she is also working on other mechanisms of the virus. More generally, she works on a variety of other parasites and disorders, including influenza virus, chlamydia, and a variety of less well-studied diseases. Among other honors and awards, Romy is the recipient of the NIH Kirstein National Service Award, the NIH New Innovator Award, and the Presidential Early Career Award for Scientists and Engineers. Romy earned both her BS in Chemical Engineering and her PhD in Chemistry at the University of Illinois, Urbana-Champaign. All questions will be fielded after the lecture during the Q&A session. Romy, the screen is yours.
Thank you very much for having me here this evening to give this special lecture. I appreciate the invitation and look forward to telling everyone all about the work that we've been doing to use computational microscopy in order to learn about the molecular piece parts of the SARS-CoV-2 virus. So some of you may have actually already heard about our work in a beautiful New York Times piece that went to press last October by Carl Zimmer. And what was so cool about this particular piece was that it really showcased, this came out in the science section of, on Tuesday in the New York Times, it really showcased the tremendous work of scientists and in particular of structural biologists to be able to visualize what the virus actually looks like, including, the virus itself, as well as this sort of the spike proteins and the molecules that comprise it. And what was also striking, so this image here that was one of the central features of their piece was an image that was created in my group. And this was in the Tuesday sort of fold out section. What was really also so wonderful about this was the opportunity to sort of bring this type of science, which I call computational microscopy, or all atom molecular dynamics simulations, which I'll tell you about, into the living rooms of people worldwide. Because, computational biochemistry and biophysics is really a very new element of these disciplines. It's relatively new because of the advent of computers and so forth. And so, so there's a lot of folks who, when they think about what chemists are, or biophysicists or biologists, you know, they think about people wearing goggles and lab coats, and they don't really even sometimes know that there are whole teams of people around the world who are, you know, who are investigating viruses and other, you know, biological systems, for example, you know, just really using computational techniques. And so that was, I think, also just a great opportunity to have through this, through that particular article. But so what I wanted to do today was really tell you all sort of the story, the more of the scientific story and the technical story behind these images, you know, that you may have seen. Okay, and so really what I'll mainly be telling you today about is a technique that we develop in my group, and you know, around the world there's other groups who do as well, something called molecular dynamic simulations. And I like to think of this approach as a computational microscope, because that's what it is, and that's how we're using it. And so nowadays, you know, scientists have a number of different ways, of course, that they can interrogate biological systems. And there's many different sort of experimental techniques that give us different angles or views of these systems. So what we can do is actually use computing to bring together different types of data sources, taken on completely different instruments in different groups all around the world, and build highly detailed, three-dimensional physical models of these biological systems. And then using relatively straightforward laws of physics, bring them to life, if you will, with molecular simulation. And so with the virus, what we've been able to do, and we've done this also for other systems, is we can combine different types of structural biological data. So this could be high resolution X-ray crystallography, also relatively high resolution cryo-electron microscopy, and tomography. So these are all structural data sets. And we can combine those multi-resolution structures together with glycomics, which is a way of, as I'll discuss in more detail in a bit, telling us about the different types of glycans that are in systems. Lipidomics, so it also tells us about the lipid content of membranes, for example, and genomics or genetics, so what type of sequences might be varying. So we combine all of this in these relatively complicated systems that, you know, can have anywhere from hundreds of thousands of atoms to now, as I'll tell you through the talk, not only hundreds of millions, but even reach out to the billions in terms of number of particles or atoms in our system. And then all we're doing is essentially we're approximating that system down to its many atoms. So we treat each of the atoms in this system essentially as hard spheres. And they're connected essentially by bonds, which we are treating as springs. And so then we define this potential function, which I'm showing here, which basically describes the interactions that the atoms have with all of the other atoms in the system. And then all we're essentially doing is integrating Newton's equation of motion over time. And so we start with some particular configuration that is specified by these data sets. And then we integrate one time step, and we get a new structure, and we integrate again, and we get another structure. And we do that numerical integration millions and billions and trillions of times. And then we can build up a dynamical understanding or a trajectory of the system's movement and dynamics over time. And so this technique runs nowadays on a number of different computational platforms, but you know, we're aided by these very large machines, the so-called supercomputers that are in the United States, but there are others, of course, in other parts of the world that are also very impressive and very fast. But the two that I will talk about using in this particular talk, in this particular application, is a computer in Texas, near the University of Texas, Austin, at the Texas Advanced Compute Center. The machine is called Frontera, and it's basically the largest the largest academic machine in the US that's sponsored by the National Science Foundation. And then a second machine called Summit, which is at Oak Ridge National Laboratory, which is a Department of Energy machine. We also use that quite extensively for this work. And so, because this is basically the engine of this microscope, these, as compute gains, have really increased dramatically and continue to increase dramatically, folks like me with these types of methods can take great advantage of this sort of growing computing power in terms of the scale and the complexity of the systems that we can investigate. And so, at the end of the day, and what I hope to convey to you is just my excitement around using data-centric computational simulations to integrate, not only to integrate the experimental data, but to give us views and insights into these systems that take us beyond where experiment can currently go. And I think that's really where we can make these, as I'll show you, very exciting discoveries. Okay, and so until about last February of last year, my group had been working in, we had worked in infectious diseases for a number of years, and we had been focused heavily actually on the influenza virus. And in mid-February, we were really excited. So February of 2020, we were very excited because this paper here that I'm showing you on the top of the slide was just published. It was February 19th, so mid-February. We were very excited. So this paper presented an all-atom molecular dynamics simulation of the outer envelope of the influenza or flu virus. And so it's shown here. We were very excited. It made the cover of the journal, and it represented about a 7-year journey where we had to develop tools and techniques, as well as perform the simulation and analyze it, all to learn something new about influenza virus. And at the time, last February, when we published this system, once it was fully built and constructed, had about 120 million atoms. And it was the largest virus simulation that had ever been simulated, and among the largest of any biological simulations that had been carried out. So it was, it presented a very exciting technological advance, while also, we were learning new things about the glycoproteins of the flu. But that same week, the world was changing. And I'm sure we all remember this. But so what I'm showing here are the total confirmed COVID-19 cases for the early months of the pandemic from January through May on a log scale for a number of different countries. And, in mid-February in the United States, there really weren't that many known cases of COVID-19. There was maybe a dozen circulating cases here, but we knew that the virus had already escaped mainland China. And we were at that point in mid-February, it was looking very grim in the Lombardy region of Italy. The virus was, really being transmitted heavily, and people were starting to get infected and sick. And so that was when our ears started to perk up, and we thought, well, maybe we need to do something here with this virus. And again, that same week, in fact, just a few days in advance of that publication, something else really important happened. And that was that the group of Jason McClelland, together with collaborators at the National Institutes of Health, published the very first cryo-electron microscopy structure, so the very first high resolution structure of the SARS-CoV-2 spike protein. And this structural information like this is really sort of one of the key experimental components of the type of work that we need to do. So as soon as we saw this, it opened up the possibilities that we could really start working in this area and try to learn something. And so this was the day that we really pivoted our efforts towards studying SARS-CoV-2. And the folks in my group who were really sort of leading the charge at that time, and at least two of whom are still involved, are Lorenzo Casalino, Ziad Ghayeb, and Abigail Domer. And it was really sort of an exciting an exciting time to be a scientist, as all of this was coming out, and sort of the ground underneath the whole world was really sort of in upheaval at that time. So let me give you a little bit of an introduction to the virus. I realize many of you probably are already familiar maybe even possibly too familiar with the virus from having been sick. But so many of us probably already know that this is what the virus looks like. It looks like a golf ball with spikes sticking out of it. And these spikes are in fact called the spike proteins. And they sit on the outside of the virus. And so they're the first point of contact that the virus has with human cells. So they play a very important role in the infection process. On top of that, these proteins, the spike proteins, are also highly antigenic molecules. That means these are that your immune system responds to them, and so these are also part of all of the approved vaccines that probably many of you have already been, if you've gotten your jabs, and maybe even now you're getting boosters. This protein is, has been of intense interest and continues to be for understanding the virus. Okay, and so what a lot of my work is this talk is going to focus on is sort of the initial infection event that happens when the virus first infects the host cell. And so the key event here, one of the key events is that, the spike protein, which I'm showing here in white surface representation, let me go back to laser pointer, needs to connect to a receptor or another protein on the human cell or the host cell. And so this protein that it needs to connect to is called ACE2, or angiotensin converting enzyme 2. And I'm showing a close-up of this interaction here in yellow. So here's the ACE2 is in yellow and the spike protein is in white. And I'm going to talk a lot about these dark blue, these navy blue bits. These are called glycans. I'm going to tell you all about those sugars. And then I also want to bring your attention to this light blue bit in the middle. This light blue bit is called the receptor binding domain, or the RBD. And I'm highlighting it because this plays a particularly important role in infection. This is the part of the spike protein that has to make contact, as you can see, with ACE2. So this is really this important handshake that needs to happen between the virus and the host cell in order to precipitate infection. So I'm going to keep coming back to that throughout the talk. Okay, and so when we first started working in this area, we thought, just coming off of that study of the flu, we really thought, okay, we would like to repeat what we just did for the flu, which took us 7 years. We want to basically repeat that for SARS-CoV-2 so that we could understand, really all of the parts of the virus in detail and to learn about how it's binding to the host cells. But when you are trying to simulate the whole virus, you don't just sort of build the whole thing all at once. We actually take sort of more of a reductionist approach. And what we do is we take each of the molecular piece parts of the virus and we make sure that we build up models of each of those pieces, and then we bring them together into the working whole. And so because the spike data came out first, and because it is such an important part of the virus life cycle, this is where we started. And so we needed to start by modeling this spike. And so the spike head actually, so as I just mentioned previously, so the group of Jason McClelland published the structure of the SARS-CoV-2 spike head first in the bioRxiv, which is a place where scientists can deposit their findings in advance of being formally published in a formal scientific journal. By mid-March of 2020, this had been published in Science. This paper has been cited, I believe, over 5,000 times in the year and a half that it's been up online. So it's mind-boggling to think of the impact that study has already had. But so this is what they presented. This is what the protein actually looked like. And so, the spike protein is actually what we call a trimer. So it has threefold symmetry, and it's made up of 3 pieces that are identical. And each of these pieces, so this is representing one protomer of the trimer, or one part of the trimer. I'm showing you here on the top. So the spike head, which they resolved the structure of, is sort of shown in bold. So the whole sequence of one of the protomers is about 1,300 residues. The spike head goes to about here. So they were able to use cryo-electron microscopy to get a pretty good picture of of the spike. And there's more that I'll talk about that they couldn't resolve. But so here, one of the things of interest is that that receptor binding domain that I told you about is shown in green in this picture, and they had resolved it in the up conformation or the open conformation. And then a few weeks after that, another fantastic structural biology group, the group of David Wiesler at the University of Washington, published another study in Cell, and this study largely recapitulated the findings from the McClellan lab for the open spike structure, but they also provided, using symmetry averaging, they provided a model of the closed structure of the spike. And so we took both of these structures, and we filled in some of the missing bits. So, experimental structural biology has undergone what they call a resolution revolution, where they were able, because of various advances in what they call direct detector technologies and so forth, they've been able to actually obtain fairly really incredibly high resolution structural data for very large molecules and macromolecular assemblies. But in spite of how powerful these methods are, they still have their limitations, like everything else. And so there are missing loops and so forth. Like there were parts of the structure that they couldn't see, essentially because they move too much. And so they can't actually resolve the image crisply enough or with high resolution. But so this is where computing can come into play. So we actually used computing to model in the missing bits that they were not able to see in their experimental biology. And we created these, what we call these full length complete models of the spike head for the open and the closed state. But now the head is just one part of the spike protein. It's an important part, and the part that really drives the initial infection event because of it contains that receptor binding domain at the top. But there's this secondary part down here, this sort of small segment, which is what they call the stalk, and also the transmembrane domain. So this spike head is actually connected to the viral membrane almost by a tether, which we call the stalk. So it's tethered to the viral membrane through the stalk. And then there's also this small bit on the inside of the virus that they call the CT domain, or the cytoplasmic tail, that sticks in on the inside of the virus. And so we used computational modeling like AlphaFold in order to create really the full length structure of this spike protein, which was the end-to-end structure. So this combined, the experimental structure with our own computationally predicted structures. Now, the other thing that structural biologists have great difficulty in seeing are these things called N and O linked glycans. And these N and O linked glycans are a very important post-translational modification of proteins. So they're things that are added onto the protein after translation, and they do all sorts of things. Like, for example, they help proteins to fold properly in many cases, or they can help with shuttling to different parts of organelle structure. But with the case of viruses, many viral proteins have evolved the ability to basically cloak themselves in an extensive sort of dressing of these N and O linked glycans, in particular N-linked glycans, in order to mask themselves from the human immune system. So these glycans are part of all the sort of many of the ordinary proteins in our body already. And so by covering themselves in this same material that is already part of our bodies, the viruses really can use these N-linked glycans as a disguise to hide themselves from the human immune system. But again, the problem is that one of the challenges with structural biology is that they can't see them. So here we can actually use computing in order to get views into their structure. So what they can do is they can use glycomics, which is a mass spectrometry type method that actually, in this paper here, this was an important paper published by Max Crispin and again with Jason, maybe in April of last year. And this paper was so important because it told us what the molecular recipe was for each of these glycans in the spike protein. And then there was another study here by Parastu Azadi at the University of Georgia that presented, again, sort of largely similar information for N-linked glycans, but also presented information about O-linked glycans, which are added to serines and threonines. So the spike protein has a few of those as well. And so what we can do with molecular modeling is actually build in those groups. So we have all of the complexity of the protein, but then on top of that, for each protomer, so for each of the 3 pieces of the trimer, there are 22 asparagines that have these N-linked glycans and 2 O-linked glycans. And so, so this is what we have, all the complexity of the protein. And then at these particular positions, we also attach this oligosaccharide unit that has all of these different linkages. And so we have to be very, very careful in making sure that every atom and every bit of this is actually correct. And I have to say, this is sort of a close-up of what that looks like. But now imagine that we actually have 3 chains of that. And so that was really the painstaking work of many of the students in my group to make sure that was all modeled correctly. And then the other thing that we wanted to model is actually the membrane. And so we knew that the viruses basically bud off of the endoplasmic reticulum in the ER Golgi intermediate compartment. And we know from many years of basic biology that each of these different sort of membranes in the cell have different compositions. Okay, so we actually can then reconstitute in silico a membrane for the virus that actually contains the correct type of composition of lipids and phospholipids. And so this was sort of at the end of the day, what our membrane contained. And so these are just different sort of very complicated names. POPC and POPE and POPI and POPS are different types of lipids in that membrane. And then it has cholesterol at 15%. And really, why we care about that is because, membranes tend to be a little bit more, they're difficult to study. We have a lot of tools to study proteins, but membranes are just challenging experimentally. So, but we do know that they're very important. And there was this, so modeling the full complexity is something that we can do in the computer, and we should do. And there was this really beautiful study by some HHMI investigators that basically showed how cholesterol, and this has now been published in Science by Cliff Brangwine, that cholesterol actually played a very important role in the ability of the virus to actually invade or enter the host cell. But so we can, and I'll show you more on the membrane later on, but there's this whole additional dimension to biological systems of membranes, cell membranes and viral membranes that, we know they're important for various reasons, but it's really it's in some sense understudied. But these types of tools that I'll tell you, that I'll talk to you about today, allow us to really view the membranes, with atomic resolution. And so, again, it presents additional opportunities for, understanding of this system in ways that are unique from experiment or complementary to experiment. And so, okay, so back to the spike protein. So what I'm showing here is actually a slice through the viral membrane that we modeled. And so, as I mentioned, so in pink here, these are in pink and in yellow and orange and fuchsia are those different lipids and phospholipids that I told you about. And this is the transmembrane component of the spike. So there's this bit that goes through the membrane. Now there's one more post-translational modification that's actually on some cysteine residues. So I told you about that cytoplasmic tail, which is this bit of the spike protein that sits inside the virus. There's this these cysteines can be palmitoylated, and these guys here, they look like caterpillars almost, but these are just basically hydrophobic groups that snorkel up into the membrane and help anchor the spike protein in the viral membrane. And so our model also has those. So this is, really this is really, we view this as the most complete structure really possible that one can build, combining all types of experimental data. Just to run through the structure again, we had the full length structure of the protein. We have it in the up conformation as well as that down conformation I was telling you about. We have all of the N and O link glycans according to the glycomics experiments that they had. You can see it covers the whole protein. We have the viral membrane with the correct composition. We have the palmitoylated cysteine. And this is what the spike protein actually looks like, with all of its decorations and all of its elaborations. That's the membrane at the bottom here. This is what it looked like. So once we had this built, I'll say we also basically dunk this protein in water. So there's also all of these water molecules and other ions that are part of something that's supposed to be sort of like a cell buffer, a solution buffer. That I'm not showing you those because if I showed you the waters, you couldn't see the protein. So I'm hiding all of those, but they're also there. And then we run our molecular dynamic simulations. And this is what the output of those simulations looks like. So here you're seeing, like Feynman said, you're seeing the wiggling and jiggling of the atoms, which can really explain to us so much about not only the dynamics, but more interestingly, the function and the structure of this amazing system. And so you see the wiggling and jiggling. And you can see too, so the spike protein is shown in gray, the sort of gray cartoon. The RBD, that receptor binding domain, is in light blue at the top. And you see these, the colorful bits, which they sort of look like ornaments coming off of a Christmas tree, right? Those are those N and O linked glycans. And you can already see, just based on this short movie, which is sort of trying to encapsulate about 1 microsecond of simulation, you can see how much they're moving around. And that's one of the reasons why they can't snap a very crisp experimental image of these. It's just because they are in fact so mobile, which you can so clearly see here. So this system of the spike protein had about 1.7 million atoms. We were running these numerical simulations, these molecular dynamic simulations on the TAC Frontera supercomputer using the NAMD2, which is a molecular dynamics engine. And we were able to get about 60 nanoseconds per day running on 256 nodes of Frontera. And so what happened was when the virus first struck and we pivoted to start to work with this system, we knew that we were going to need compute time in order to do that. So we turned, I called up, I walked down to my postdoc's office and I told them the structure was just released, like we should start running with this. And they started working with it. And I walked back to my office and I called up Dan Stanzione, who's at the Texas Advanced Compute Center. He's the director there. And we had worked together for some number of years, as I had mentioned, with influenza and other systems. So that we knew each other. And I told him, I said, Dan, there's this virus is not looking good. We think we want to try to do something with it. And he, at that point in time, people were really realizing that we were all in trouble here. And he basically said, Romy, we're going to give you unfettered access to this machine. You just, we know you're doing, you can use it. And then we'll figure out the accounting later. So they were very gracious, and in fact are still just have been tremendous. And I'll also discuss this again later in the talk, how important the response was from a number of not only government agencies, but also industrial players and so forth, people who had computational resources who gave them. And that was really fantastic. And so what that allowed us to do was not only to build the system, but then to simulate its dynamics and to really explore it without interruption. And so in just a matter of weeks, we were able to develop or sample, multiple microseconds of dynamics for this system. Okay, and then one more thing I want to mention is that is really important is that you see this image, people see this image, and what you see is I hope it's an aesthetically pleasing representation of the of the system. I mean, it's a pretty picture and a pretty movie, and I'll show you more of these, but it's really important that I convey to you that these simulations are more than just pretty pictures, and they're more than just video games. We have to always remember, like I told you at the beginning, molecular dynamics simulations are numerical statistical mechanics. And that means that, you know, we are simulating these equations, the mathematical equations, and doing that in a way that is theoretically rigorous. So in accordance with the laws of statistical mechanics. So yes, that means these calculations are quite computationally expensive. But as I hope to also to convey to you and to convince you that this expense is worth it, because at the end of the day, what we can do is extract back out from many averaged microscopic properties. We can extract back out experimentally testable macroscopic observables. Okay, so that's a bit of a mouthful, but really what it means is that these are more than just pretty pictures, that we can compute from these trajectories. We can compute properties that link directly to experiments that they can make in the lab. Okay, and so this is the real power of the approach, is in using these simulations to help drive, well, what is the next experiment to do, and to make predictions that they maybe can't even measure directly, but that can be orthogonally verified. So again, hopefully this is something that comes out when I start to talk about the results. Okay, so let's get to the results. So what did we learn? So one of the reasons why our work was so well received was because we showed the world for the first time what the full length, fully glycosylated spike protein of this virus actually looked like. So on the left, what I'm showing in light blue is the spike protein, essentially as what the structural biologists see. So they can primarily just see the protein. They can see the individual, there's some sugar linkages that they can see that first sugar unit, but they see the protein. And this is the membrane. On the right is what the spike protein actually looks like. If they could see the glycans in the experiments, this is what they would see. You would see something that really looks quite different, right? We have, you can see, so each of these dark blue tufts, or tufts, if you will, are basically, this is representing one glycan. It's a composite image where I'm showing many snapshots of the protein overlaid over one microsecond of dynamics, okay? And so, like, for example, this like puffball here is basically all of the different structures that one N-linked glycan, all that it occupies over a microsecond of dynamics. So what you get, hopefully I give you the sense that each of these glycans that you see on that structure, they're moving around a lot, and they're basically sweeping out a whole sort of like region of space. And so collectively, they basically form this like dynamical coat or dynamical disguise, if you will, that literally you can see so clearly here. It is hiding the foreign agent of the spike protein, which is foreign. You know, we don't have something like this in our body natively. If what our body saw was this, it would be able to immediately recognize it and you know, detect it, and your body would respond to it. But instead it sees this, and these blue bits are just like what it sees everywhere else. So it really is a very effective disguise. So that was cool. And you know, this type of information was really critical. It is critical for vaccine efforts and also for drug design efforts. In particular, you know, if you're trying to develop a therapeutic, you want to, maybe it would be possible to design a small molecule drug that would bind to some particular exposed part of the spike protein and maybe stop it from working, for example. So there were a lot of folks that were interested in this. Our simulations not only showed the shield, but they also showed why the spike had these two different conformations from the experiments. It actually explained why the spike why it opens and closes. So on the left, what I'm showing is the closed spike structure. This is sort of from the top view looking down. And what you can see is the receptor binding domain, which I've been showing in light blue, and I'm going to continue to show in light blue. You can see how it's sort of hidden, it's obscured a bit by these glycans. And if you look from the side, you can see it's this light blue bit. It's basically that receptor binding domain is sort of tucked down beneath the glycan shield. But this is if you contrast this to the open spike, now again, this is the top view looking, down on the spike and from the side, you can see how it's when it's in that up conformation or that open conformation, it presents a completely different view. Now the receptor binding domain is completely exposed. And look how dramatic this is from the side. You can see so clearly how this protein has now moved this part of itself that must make that handshake with the host cell receptor. It's now up above that glycan shield. I just think that's so amazing. So, this is really, if I like to think that if the structural biologist could have seen the sugars, instead of calling it up and down or open and closed, they would have called this like the defending mode and the attack mode, because in one case it's really shielded, and in the other, now it's sort of lifted up and can actually infect the host cell. So the other thing that we can use, or one of the other sort of reasons why our studies can be useful and interesting, is because we can also explain how these different neutralizing antibodies are binding. So there have been a number of neutralizing antibodies that over the course of the past year and a half have been published that bind to the spike protein, and can actually neutralize the virus. So I'm showing here sort of the footprint of binding locations of different neutralizing antibodies that have been structurally characterized for the early months of the pandemic. So, now we have hundreds of these that they know about. But so there's that, when that spike protein goes into the up conformation, that part that binds ACE2 is something that is recognized by our immune system. And we make antibodies, basically against that, recognize that part. It's antigenic, it's the epitope. So you can see there's a number of those where they have binding here. There's this one called B38 and another 47D11. There's a whole bunch of those. But there were other neutralizing antibodies that were binding to epitopes or regions of the spike protein that were sort of harder to understand if you just looked at sort of the open and the closed conformation. And so our types of models can be useful in understanding how those different molecules are sort of binding in these complexes. And so this was also something that folks at various companies and so forth were very interested in. The other thing that we showed was how glycans not only are part of the shield, but that glycans actually can be a way that the virus escapes the immune response. And so we worked together with Rino Rapulli and Jason McClellan to model what happened to a lab passage strain of the SARS-CoV-2 virus. So Rino and his group basically cultured the virus in the sera of a patient that had many. It was what they call a potent patient polyclonal sera. So it has a patient that has already developed neutralizing antibodies to sort of like the wild type spike, or the first type of spike that it saw. And when you do that, basically, these viruses continue to mutate over time. We know this now, probably, I'm sure people have experienced it with Delta, for example. It's basically increasing its fitness as it continues to circulate across the globe. And so, one of the reasons why we really do need to try to keep infections down is because every time someone essentially is infected by the virus, we give that virus another shot on goal to escape immunity from our current treatments and our prevention measures. So in any case, so what they showed was that over time, when they cultured this, when they did this lab passage strain of the virus, after 90 days, it was in fact able to completely evade the immune response. So it was able to essentially it would be able to reinfect a patient because it changed so much that the defenses that we had are no longer viable. And one of the real sort of kickers here, or one of the things that the virus did that provided a huge fitness advantage against, or really provided the immune escape, was that there was a very large deletion in the N-terminal domain. And not only did it was like a large loop, which presented a big remodeling of that site, which is important because if you have remodeling, then you destroy the ability of two proteins to come together. But it also introduced a new glycan sequence. So it not only added sequences to itself, but it also inserted a new sugar molecule, a new N-linked glycan. And so here, what I'm showing in this upper right panel, I'm showing the side view of this part of the spike actually bound to a neutralizing antibody in gray. And in yellow is this insertion, and in dark blue would be if the glycan was there, as it was after being lab passaged. You can see that when it inserts this glycan, because these glycans are these large oligosaccharide chains, by inserting something right where a neutralizing antibody would bind, it basically obliterates the ability, it eliminates the ability of the virus then, of that neutralizing antibody to bind. And so that's, really one of the main, mechanisms of immune escape. So this showed like another role beyond shielding for glycans, that they can also be used in immune escape. There we go. But then we even noticed something else. So when we were looking at these models, and when we looked at and this I have to give a special shout out to Elisa Fada at Maynooth University in Ireland. She's a fantastic glycochemist, glycobiologist. We actually met on Twitter. So I have to say, I never did social media before the virus, and I joined essentially Twitter just to sort of stay up to speed with the science of SARS-CoV-2, because it became a way for scientists really to communicate findings instantaneously, around the world. It wasn't that, you were waiting for a journal or waiting for emails, and we were all sort of kind of we were stuck in our houses, right? There was lockdowns happening. So we weren't having conferences where people were presenting unpublished, work. Twitter really became a place where scientists would very quickly and rapidly exchange information and data on, on their experiments. So that was interesting. So I ended up meeting a lot of people, as I'll talk about, but also Elisa was one of them early on. And when we were looking at the spike protein, we noticed something. This is the closed structure of the spike. I'm looking at the top-down structure, and again, you can see that threefold symmetry. So there was one particular glycan that was always sticking sort of outward in solution. So the glycan here is shown in the sort of van der Waals spheres, or they look like balls. It was always sticking out. Now, when we looked at the open structure, we noticed that two of those glycans looked normal, but one of them actually was pointing inwards. And so we took a closer look, and now I'm showing a different view of the spike protein. So that receptor binding domain is in blue, try to keep it in blue throughout. And now the other domains of the spike protein, those other trimers of the top, the spike head, are shown in red and gray. What we saw was something very curious. It was that when we looked closer, there were two glycans in particular, one at position 165 and one at position 234, that when we rebuilt the glycans into that up state structure, they actually filled up the void space that was created when that RBD lifted up. And so it looked to us as if these two glycans were sort of propping up this receptor binding domain, sort of like a kickstand on a bicycle, right? That it was giving structural scaffold to the protein. So we came up with essentially the hypothesis that maybe, that maybe these were actually providing structural support. And so we mutated in silico, so in our simulations, to test this hypothesis, we mutated these asparaginins to alanines. And we did this because they can also make this experiment in the lab. And I'll tell you about, they did do that. But so we could already in silico start to explore these experimentally testable hypotheses. And so when we eliminated these glycans, what we saw, then we ran more dynamics. What we saw was this receptor binding domain actually destabilize and collapse downwards. And so that basically supported our hypothesis. And so then I called up Jason McClellan, and it was funny because he was someone who I had never interacted with before, but I had posted a movie of the spike fully glycosylated, and in Twitter, and he replied to it, and I thought that was pretty cool because of course everybody knew who Jason was because he had made the structure available. He was the first to have the structure, which was such a big deal. And he also, it turns out, is co-inventor of the Moderna vaccine, and has made really seminal contributions to the vaccines themselves, in the sense that he was really the lead author. There were others involved, but he was, of course, but he was a lead author on this so-called 2P spike mutant. And so I don't want to go into too long of a sidetrack, but I will say that this is a very important mutation. So he and some others had shown for the MERS spike some years ago that actually when they make these 2-point mutations on what they call the central helices of the spike, so inside the spike head, what happens is it stabilizes the spike in that 1-up state. And this was really important for 2 reasons. The first reason was that when they stabilized that spike in that 1-up state, it all of a sudden allowed for the cryo-electron microscopist to determine the high resolution structure. Because when they took all of their images, there was one particularly well-populated state of the 1-up, and it was enough to get a high resolution structure. But the other reason why that was important was because when that spike is in that 1-up state, that highly antigenic part of the receptor binding domain that I've been telling you about, the part that needs to make that handshake with the ACE2 receptor, is exposed up above that glycan shield. And so not only then does he stabilize that spike so that it can be structured, it can be studied with structural biology with cryo-EM, but also they had neutralizing antibody titers that were like 10,000-fold what they had seen without that mutation. And this mutation is in the Moderna spike, and it's in the Pfizer spike. It's basically in all of the vaccines, I think, except for the AstraZeneca formulation. And so that's one of the reasons why it's also been such a, I think, our bodies are so responsive to it. So anyway, so sidebar, but so I thought he would be very busy, but it turns out, that he was just fundamentally interested. He's an amazing scientist and is fundamentally interested in understanding this system. So he said, "Yeah, Romy, sure, we'll make the mutations and we'll test it for you." So Jason and 2 folks from his group, Jory and Christy, created a new type of assay. It's a bilayer interferometry assay, where the 2P spike was the positive control. And then this gray line here is a negative control, where they basically engineered a spike that has a disulfide bridge. So it basically used chemical bonds to basically lock it in the closed conformation. And so this guy never interacts with ACE2. But basically what these traces are showing are the interaction that the spike protein has with that ACE2 receptor in the host cell. And so when they make these mutations of N165A and N234A, they see this reduction in interaction compared to wild type. And they couldn't actually make them both at the same time because there were issues with the expression, but it was enough to demonstrate or to support our hypothesis that that these glycans at N165 and position 234 were actually playing a role in infection. And so this was really cool because it had never been shown before for viral proteins that so people always think about this sugary, these glycans as the shield and the shielding, the glycan shield, the sugary shield, but they had never actually been shown to participate in the mechanism of infection. And so this really opens new possibilities for our understanding of glycans, and that not only was it a shield, but part of the weaponry itself. Okay, and so all of that was really looking at these two states of looking at relatively short time scale dynamics for biology, microseconds of dynamics of the spike in the closed state and then in the open state. But what we really want to do, if we want to understand how this protein works, what we really want to do is actually simulate sort of the opening transition. And then, if we could, it would be great to actually get atomic level views of, the whole thing opening up and the fusion of the two membranes, coming together. But that is very difficult. That's a very long time scale event to be studying with simulation. So we can't, it's actually relatively intractable with standard or conventional molecular dynamics. But so we then partnered with Lillian Chong at the University of Pittsburgh, who's a fantastic theoretical chemist. And she develops a method called weighted ensemble molecular dynamics. And so what happens with these simulations is a lot of time you basically are sampling these small motions in sort of these valleys of the energy landscape. So this is a picture of the energy over some progress coordinate. We spend a lot of time sort of waiting around for things to happen. Her method is a statistically rigorous way of enriching sort of the uphill, sort of helping to these molecules to sort of get over these interesting barriers, these interesting functional barriers. And so what it allowed us to do was actually, I gotta go back to change my thing here. What it allowed us to do was actually, oh no, it didn't play. Oh, there we go. Oh no, it's not playing. I apologize for that. Okay, well, maybe I can drag it. It basically allows us to study the opening process. So we start with that closed spike, and we are simulating the spontaneous opening sort of it. So we're not adding any forces. We're just using her method. It basically helps us to actually view the entire opening pathway. Let me see if I can play this movie. I'll just drag this bar across. So, so here it's closed, it's closed, it's closed. Things are wiggling and jiggling, wiggling and jiggling. And then all of a sudden it opens up. All right. And so, so it basically allows us to go, oh, there it's going to play. Oh, just for a little bit to the open state. Let's see if it plays. No, it's not going to. That's okay. Don't worry. There's more movies. So, yeah. Okay. So, so what's really important about this, again, so let me just reiterate. So if we wanted to actually just use conventional MD, like I told, molecular dynamics, like I told you about, and we wanted to sample that opening transition, if we wanted to do that once and just using conventional molecular dynamics, it would take on the order of years to see that. And that's because the barrier, we know that now from different experiments from the group of Walter Muths, that basically this opening process, it happens on the seconds time scale. And our molecular dynamics simulations, that time integration step that I mentioned at the beginning, I forgot to tell you, it's really small. It's basically 1 or 2 femtoseconds, or maybe as much as 4 femtoseconds, but it takes us a long time to get to the seconds time scale. So it would take us years to do that. But with her method, with weighted ensemble MD, in a matter of weeks, we were able to generate hundreds of these opening pathways. Okay, and we did that we did that by taking over. So we run in parallel many different simulations on these individual graphical processing units, or GPUs. And so, so it turns out one of the really great things about molecular simulation over the past several years has been that the same types of chips, so computing chips, graphical processing units that they use to, render amazing video game graphics, which is a billion, I don't even know how many billion-dollar industry. Those same chip architectures map very well to the scientific problems that we're trying to solve. So we can use those same. So GPUs now are sort of like bringing supercomputers into the houses of people worldwide, which is really, really cool. And anyway, it's been very powerful and allowed us to learn things about the virus and other proteins that we otherwise couldn't access with like CPU type technology. Okay, and so something else that happened just by chance. I was talking with Joachim Frank, who I believe Larry was telling me actually gave one of the last seminars for this group. He's at Columbia University, the 2017 Nobel laureate in chemistry for his sort of seminal work in cryo-electron microscopy. His graduate student, Evan Seitz, who's fantastic. It turns out that they had actually taken Jason's data set, and they had been trying to work with that data set to understand some of the dynamics of the spike protein. And so Joachim and his collaborators have been developing something called Manifold EM, which I'm not sure if he talked about. But it basically, so what typically what happens with cryo-electron microscopy is that they're very focused on getting this high resolution structure. And the way they do that is they take like hundreds of thousands of images of the molecule, and then they bin them and they group them up, and they try to they collect enough. They try to get like a lot of images of the molecule in one particular state, and that's what gives them their high resolution structure. And they throw out, generally, they literally basically throw out all of this other data that basically, might have useful information about other states or dynamics that these systems are sampling. So he's been developing with collaborators something called Manifold EM, which attempts to connect the different states that they see in the cryo-electron data sets, essentially to an energy landscape. So in order to sort of like understand, to take better advantage of these rich data sets beyond just getting the single structure, like trying to understand different snapshots or different configurations that these systems might sample over time. So what they had found was that, so basically they had found when they analyzed the data sets, now show you. So what I'm going to show you on the left is our weighted ensemble trajectory. But now we've just made it look like what they see, because we want to show you like this is what they see from cryo-EM, and this is what we see, but we've made it to look like what they see. So we've reduced our resolution to match theirs. And what you're going to see is our opening trajectory, what we see, and then also what they saw collected out of the experimental data sets. So there's our weighted ensemble trajectory. And I'm going to play that twice. And here you can see what they see from the cryo-EM, and it's very similar. Let me play that again. So what they had already sort of gotten the sense of when they looked at the other structures in that data set, Jason's original data set, they already had the sense that there was something happening. But Jason never talked about that because these are not high resolution structures, right? So that was really what his focus was on. But it was useful for us because what it showed us was that our computationally derived, theoretically derived opening motion actually aligned very well with what they had found in an actual experimental data. So it gave us some nice sort of validation that this motion that we saw was really backed up actually by structures that hadn't been seen yet. Okay, and so here again, I'm showing, this is now the movie that, a different view of the movie that didn't play so well earlier. This is the top of the spike protein. The receptor binding domain is shown in blue, and it starts closed, and then it goes to the open conformation. And I just want to also say, there it goes, it opens up. And one of the things that was so interesting about this was that I'm highlighting here this glycan at position N343 in hot pink. And why that was so interesting was because when we just did our original simulations where we were looking at a sort of short time scale dynamics from around the closed structure and around the open structure, this particular glycan here, this 343, it doesn't look like it's doing anything interesting. In both of those, if you look at the two end states, the open and the closed, it just looks like it's sitting on the side, not doing anything. It's only when you actually look at this opening pathway that we saw that actually this glycan was doing something. And in fact, it was doing a lot. It was basically acting like a gate that helped to swing open that RBD and then actually lift it up, which was really cool. So we went back to Jason and we said, hey, we have another hypothesis that maybe, you know, this glycan is important. And they went back into their experiments and they created that mutation as well as some other mutations of residues that we thought were important. And they actually found that this glycan was even more important than the other two in terms of like reducing the ability of the spike protein to interact with ACE2. And this work was led by Tara Stain and Shirley Ahn, who are fantastic folks, scientists, and was published in Nature Chemistry a few months ago. Well, and so, you know, beyond the spike, of course, what we want to do is actually study how it's interacting with ACE2 and other, you know, we want to understand really what the virus is doing when it gets to the cell. So we had also studied the ACE2 receptor. And so here they also had structures of ACE2, which is the human receptor, and it also has glycans on it. This is what that system looks like. This was actually built by Amelia in my group. And what we found here was that this receptor was also really flexible. And it was, I'd say, surprisingly flexible. We actually saw this guy, and you'll see it. It basically, you know, it moves around, and then it actually can tilt back and forth and almost go completely parallel to the membrane and actually touch it. So we saw this really sort of like wide swinging motion of ACE2, which was unexpected. And we sort of, you know, we hypothesized it's actually, you know, when the spike protein, it binds to ACE2, and then it's that connection between those two molecules that basically helps to bring the two together long enough such that then the two membranes can fuse. And that fusion process is really modulated by these larger scale changes in the spike protein, which I don't show you any pictures of here. But it's this rather large sort of mechanical process. And so it makes sense that these different proteins have these flexible joints to help facilitate that mechanical process of host cell fusion. And so we then sought to bring these two systems together. So we have the full length spike, which we had built up in the open conformation, and we brought that together with ACE2 to create a system where we could actually start to look at the atomic level of this interaction, you know, in its sort of complete form. And these are just sort of technical details, which I won't bore you with. But this system now had, so the spike system, as I told you, had about 2 million atoms. This system here has about 10 million atoms. So it gets a little bit bigger. It's a little bit harder to deal with in terms of the complexity and more expensive to simulate. But we did do that. And so here is actually, this is the dynamics of that system. And you can see, you know, here's the host cell membrane and the viral membrane. So this really gives us this highly detailed picture of this initial interaction event, where there's a lot of things that we can learn about that contact. But then also, interestingly, it allowed us to actually look at other elements of the spike protein that people paid lesser attention to. One part of that is the stalk part, where they can't generally resolve that with high resolution cryo-electron microscopy. But we saw a fair degree of actual bending of the stalk, which was really interesting. And actually, oh, this is the movie that didn't play before. Look at that, it's playing in this one. So this is that movie, the weighted ensemble movie. So one of the things that we're trying to do now, that many folks are trying to do, is use artificial intelligence to help us carry out these simulations more efficiently. And so we partnered with another team, Arvind Ramanathan at Argonne National Lab at the University of Chicago. They've been developing something called Deep Drive MD, which basically uses an adversarial autoencoder. It basically learned about the dynamics that we had developed using the weighted ensemble simulations, and then used that information in order to help drive the dynamics essentially of the spike protein in this sort of like larger and more complicated scene. And so it helps us. It's a way of it's a deep learning based method that basically helps make these simulations more efficient and sort of help go to more interesting biologically relevant places. Again, because one of the things that we struggle with is that we have all these atoms. And it takes a lot of time to do the computing, and it's just we want to see the interesting events, not just wait around. So this method also helps us do that. So this is what so this is another view of that interaction of the spike with the human ACE2 receptor. And this is a schematic. So, you know, I had talked a lot about the spike head and about the interaction of the receptor binding domain with ACE2, but there's this other part down here of the spike protein. So we call this the head. And then the stalk has something like a hip joint and a knee joint and an ankle joint. And they really are like joints. And so we can, with these simulations, basically study, like here's, we're looking at how the ankle moves, and we can look at also how the knee moves, and we can look at how the hip moves, as well as the, you know, as the head relative to, you know, to the membrane. And so, you know, we do this. This is interesting because, you know, first of all, because this is impossible to like structurally, almost impossible. Well, the dynamics are impossible to structurally characterize, experimentally characterize. So it gives us a view of this, but also again helps us understand, you know, how this protein can actually facilitate the fusion of the host cells. And so then beyond looking at those two interactions, you know, the interactions of those two species, so the spike protein and the ACE2, we were also really interested to try to model the whole virus, as I talked about. And so last summer around August, there were 4 different amazing structural biology groups all over the world who were also racing to be the first to publish cryo-electron tomography experiments of the virion structure. Okay, and so this is just a different type of microscopy, which basically gives pretty high resolution information of the spike protein, but in the context of the whole virion. So it gave us information about how big the virion was, like in terms of its membrane envelope, or its lipid envelope, as well as the number of spike proteins that each virus would have, and the distribution of the spike proteins on the surface of the virus. And also what they saw from their tomography images actually was reminiscent of what we saw in the spike simulations with H2, where they actually did see that there was a lot of bending of the spike around these different joints. Okay, and so you know, once we had that experimental tomography data, then we were very interested to build up the whole virion. And so to do this, we actually have to build an all atom membrane for the virus. And this is actually really the hardest part. I would say the biggest obstacle to simulating at this scale is generating a numerically stable membrane. And that's because of that there's differences in the curvature of the inner and outer leaflets of the membrane cause all sorts of problems for numerical stability. Because in these simulations, you know, you have millions of atoms, and if any one of those atoms, if any 2 atoms get too close, essentially the energy just, it goes through the roof. So the simulations become unstable. And so we had worked, and actually this was really the hard work of Abby Domer and my group, to develop sort of an iterative protocol that helped us build and model the virus in the membrane. And so here you can sort of see it coming together. So first we start with like this relatively straightforward ball of lipids. And then as we simulate it, the different densities are sort of equilibrating. There's holes that form and clashes. And so we put, you know, we have to sort of fix that, smooth it out, and then we put in the spikes. And at the end of the day, we were able to actually construct and simulate the virion in spikes in their sort of in the virion system. And this was run, we got this up and running last October on the Summit supercomputer. And so this system of the virion now has 300 million atoms, and it was the biggest system. Certainly, it was set a high watermark for computational virology, and was part of this larger effort for the Gordon Bell Prize. So, so this is representing sort of all the work that we had done starting in February from those first images of the spike. We really sort of rapidly mobilized an international team. These are their names, and I'll show you their pictures in a moment. To do everything from studying, the spike protein with its sugary shield, to understanding how it's opening, to developing these adversarial autoencoders so that we could study the dynamics of the system in this more complex scene, and then also to finally build and simulate this 305 million atom virion. We did this in a matter of months, which was just, incredible. And one of the coolest things about this was, not only the science that we accomplished. Oh yeah, and here's my first tweet in March. That was crazy. That was when I met Jason, because he saw that and he thought that was a pretty cool thing that someone had done with the structure. So that was how we started interacting, which was neat. But in any case, this was, it was just such a special time, and it still is really, but I mean, those early days, it was such a special time to be a scientist, because, and especially a scientist in this space, because, we just wanted so desperately, I think all of us did, to try to do something useful, in this new challenge that sort of faced, humanity. And this was one way that we sought to do that. And so, this work, we learned many things scientifically, but also broke a number of new ground technically. And so we submitted that paper, and we ended up, winning it, which was really cool. And this is the team. So it was the work of a 28-member team that involved 4 different supercomputing sites in the United States, as well as a friend in the UK. But it was just phenomenal. And, a lot of the work, we talk about all these technical parts, which are really important, of course, but, a lot of it also comes down to people, and so I showed that picture of the team, and it was so important to have people come together. And, I think it's so fantastic how you kept your lectures going, on Zoom. We also, as scientists have, we pivoted really strongly and created all these wonderful, close partnerships with people all around the world, in part because of platforms like Zoom, which allowed us to connect really seamlessly, which has been really fantastic. And, early on when we started talking and working on this, we realized that we really had to do something different, that this was unlike anything that we had seen before, because, scientists, we tend to be, we hold our cards very close to our chest. We want to be the first to discover something. That's really where you get that you get that science paper, you get all the acclaim to be first. And you still do, of course. But when it came to the virus, it was more just that, people cared less about that. And it was more about, like, we really need to try to help in some way, and to try to develop drugs, or help with a vaccine, or just learn about it. So we recognized early on that we need to do something differently. And so myself and other colleagues, there were over 200 molecular simulation groups who signed on. We developed a shared set of principles that helped us to just committing to sharing data openly, as well as our methods, and to share it as early as possible, to adopt the use of preprint servers, and to be really open with with everything in order to advance our understanding of the virus as fast as possible. And you can see in the science that has fallen out of that, not just in molecular simulation, but also just in general. This was sort of like the way that people, the way that scientists responded to this new challenge has just been fantastic. I mean, phenomenal what we have learned and what we have done. I think, if there were any silver linings to this pandemic, which is in some ways difficult to think about considering how many millions of people have now died, I think science really sort of showed up and did something wonderful there. So that's good. And so we also developed a new way to share our data. So the other thing that we're not so good at molecular simulation, or we're challenged to do, is to share our data, because these data sets are really big. And they're sort of messy, but we tidied them up and created a framework where we could share data, which is still growing and still very important. And this was really supported by the Molecular Software Sciences Institute, which is a very large investment by the National Science Foundation to do that. The other thing that was really important was that for decades, the federal agencies had invested funds to support the development of these simulation and molecular modeling tools like VMD and NAMD, so that people could freely use them. So we don't have to pay money or software rights or software licenses for the code. So everything that you see in terms of the software was freely available and really high quality software. And so that's sort of just saying the importance of continuing to invest in science, and even when it seems kind of basic, that this is something that helps us be more prepared, of course, for future pandemic and future threats, regardless, could also be with climate change or energy and water, et cetera. And then the other thing that I touched on was really something that was really just, again, so important, was how people, scientists wanted to bring their expertise, but of course there were people who had resources to give, for example, like the compute providers. And they formed something called the COVID-19 High Performance Computing Consortium, which this was academic sites, but also industry sites, and so Microsoft and Oracle and Google and the Department of Energy, and all the folks said, they came together and they formulated a way to allow people to get access, for people who were studying COVID-19 research, not just for the types of models that I showed you, but also for epidemiological modeling and agent-based modeling and drug discovery, that they could actually have access to these computers, and rapidly. And that's the other thing. So it would have been literally unimaginable to think that we could have even gotten access to these machines to run all of the simulations that I told you about without the support of these folks, without them saying, yes, you can come in. Because otherwise what scientists have to do is we go through a rather long application process where, we write something up. It's a 15-page document that then gets sent, it sits there for a while in a bureaucratic pile, and then, it gets reviewed, and then 6 months later it's awarded. So it takes at least a year usually to get access to these systems. These folks figured out how to do it when it really mattered. They could do it in a week. And so that was fantastic. And now this will be a legacy from this pandemic. They've actually codified this HPC Compute Consortium to now more rapidly mobilize, when another future threat comes. And I'm sure it will, and because these things will all be accelerating because of climate change and the intersection of that with human health and so forth. Okay, so that was basically what we did last year. In the last few minutes, and I think I hope I have a little bit more time, although I'm going really long. I want to talk to you about what we're doing now, which is, I think, I'm even more excited about. So, a lot of what we have seen with this virus, there was a lot of misunderstanding and mismessaging about whether or not this virus is airborne, and we know it is airborne. We know that now, and but it took a long time for folks like, for example, the CDC and even the World Health Organization still today refuses to use those words to say that it's actually airborne. For what reasons, really, honestly, I do not know, because it is so clearly airborne. That's why masks are the most effective way of preventing disease transmission. In fact, they're more effective essentially than the vaccines themselves, and why we need a layered approach to interventions. But in any case, so we know that COVID is airborne, but one of the challenges to really understanding that is that the study of aerosols, and so aerosols are these sort of, they are these, I think I'm going to show a picture of this next. So the aerosol, when we cough or when we talk, we are emitting not only droplets, which are sort of these larger particles that you can almost macroscopically see, but we also create aerosols, and the viruses are inside these aerosol particles. And so in work that I didn't tell you about, my group had been working with a center here at the University of San Diego, or University of California, San Diego, called CASE. It's a very large NSF Center for Chemical Innovation. And CASE stands for the Center for Aerosol Impacts on the Chemistry of the Environment. And so in CASE, we had been working on trying to understand something called sea spray aerosol, which is when waves crash, they create these sort of like bits of the ocean that get aerosolized. And these aerosolized bits of the ocean basically go up into the atmosphere and troposphere, and they do things like catalyze all sorts of chemical reactions. They do things like seed clouds and, cause ice nucleation. The folks at CASE who were aerosol experts, in particular Kim Prather, were really trying to convince people that the virus was airborne. And so they've been doing a lot of work trying to get the word out and get the message out about the importance of acknowledging that it's airborne. Because once you acknowledge it's airborne, then you can create these mitigation measures like the ventilation, air filtration, and the masking and so forth. And they had a whole bunch of, or not a whole bunch, but there were several very high impact publications on airborne transmission in Science and The Lancet going on in parallel to us, building the virus. So what we are doing now is we're intersecting our work on aerosols now with the virus. And so here I'm showing CASE investigators also have pivoted a bit from studying sea spray aerosol to studying the aerosols coming out of people, of infected people. And so here you can see, this is from one of Kim's papers that has been downloaded. This paper in Science has been downloaded, I think, more than any other paper in the history of the journal. And it's basically showing, someone who, when they're speaking, you're emitting all sorts of particles. You have these larger particles called droplets that basically, by droplet physics, they basically drop. But then you have these other particles, these aerosol particles, which can be very small. And here's the particle size going from 0.1 micron to 100 microns. This is the relative scale bar. These very small particles that can stay suspended in the air for hours, up to 12 hours in the air, minimally. Actually, aerosols can travel around the globe. That's a talk for Kim to give, and maybe I guess you might hear her talking later in the spring. But so what we wanted to do is, now we had this virus, this virion that we built, and now what we're doing is we're putting that virus into a respiratory aerosol particle. And so these respiratory aerosol particles, they don't just contain salty water, they contain all sorts of interesting species from our bodies, I guess, obviously, but they have things like, they have different lipids, they have cholesterol, they have these things called mucins, which I'll tell you about, and proteins. And so that's really what we're working on now. And so, these simulations are, so this is a picture of the virion, and this was what we did last year, which was really groundbreaking. We now have these billion particle systems of the respiratory aerosol. So it just, to give you a scale bar here, so this aerosol is about 300 or so nanometers in diameter, so 0.3 microns in diameter, and they have the virion or the virus inside of it. And so they have their composition, as I mentioned, can be quite complex, and we can basically tune the composition of these aerosols. So the ingredients that we put in them, we can change those ingredients and match experiments, so we can really create this this sort of this again sort of this really sort of virtuous cycle between experiment and theory in order to really get a better understanding about sort of what is happening to the virus in the when that's in this aerosolized form. Part of that is understanding something called mucins. And so all of the interfaces in our body, that basically the air water interfaces, or like any place where, I mean, not just our skin, but like in our nose, in our mouth, in our GI tract, et cetera, are basically, all those surfaces are covered in these things called mucins. And so this is what it looks like. And these are these long, sort of complicated brush-like polymers that are coating the cells. So we needed to create models of those in order to put them into these respiratory aerosols. This is part of the ingredients of what we've been building. And again, these are sort of these long polymers that are mixtures of, it has like a protein backbone, and then again, sort of these oligosaccharide or glycans sort of coming off of it. And so we've created these models of these brush-like structures and actually simulated them so that we can include them in the respiratory aerosol. And so this is interesting. This is kind of cool because nobody has ever simulated these before, and nobody's ever actually seen their structure at this level of detail. Like, you know, because that's one of the things that we can get these pictures of biological structure. They're much easier to take when it's sort of a wide field view at lower resolution. But as you try to continue to understand where the atoms are, it's very difficult to do, to get sort of those resolution pictures with that resolution. So, so the other thing that we're putting into these respiratory aerosols is the Delta variant of the virion. So last year we had built the Alpha variant, which was what was circulating. But now we know, as I mentioned, the virus is mutated, and Delta has evolved the ability to have a lot more fitness. It is a lot more transmissible, and scientists are still really trying to understand, why is it so much more transmissible? And there are changes not only in the spike protein. So this is showing in the spike protein. This was our original study of that wild type. Or it was the strain from Wuhan. But now with Delta, there's all these different mutations all over the spike, and they're probably contributing to different features of the spike as part of what is, giving it sort of improved fitness and ability to infect. And this is just one part of the virus. There's also mutations in other parts of the virus, but so now we have built a refined virion that actually has the Delta spikes, and it's got it also has the M protein. So it's a more complete virion model. It also has these ion channels, this E protein also, which our initial model did not have. And we now have put this into a respiratory aerosol where we know the molecular ingredients of, for example, what the deep lung fluid is like. And that's basically what gets emitted in these aerosols. And so here, this is showing a respiratory aerosol particle that's about 300 nanometers in diameter. And here's the virion on the inside. And we have this with all atom models. So this is now a billion atom system. And it has mucins and proteins and all of the ingredients of the respiratory aerosol. And what is so exciting about this is that people otherwise have never seen what a respiratory aerosol actually looks like. Like, we have ways to measure their size. We have ways to determine the composition in terms of the ingredients, but we cannot see it experimentally. So this is really giving scientists, the first views of what it actually looks like. And why this is important is because this is something I try to convey to people, which it seems sort of basic, but like, when you can actually see, it's one thing to investigate something and to learn these different features of it, but when you can actually see something, it completely changes your ability to make scientific hypotheses and to understand these systems. So So I think this is really now opening a very exciting new frontier for study, where we're now intersecting virology and all of our expertise with virology and biology with aerosol chemists and aerosol scientists. And this is, again, really interesting because it's a complete intersection of fields that really kind of don't talk to each other very often. So they have, there's this whole communication challenge there in it, but there's so much to be gained, when you get into these really interdisciplinary areas of science where this understanding that the elements, the molecular mechanisms controlling airborne transmissibility, I think it's going to be a very important scientific theme, over the next few years and decade. And tools like this are going to help us, really explore these different features in detail. Right. And so, the questions here, there's so many questions of what we can do from here. I mean, one of the major questions is, so here's another picture of, somebody emitting these particles, and this is what it looks like. So, one of the major questions, the atmosphere is really destructive. You have sunlight, so you have photochemistry, you have oxidation, you have acidification, you have dehydration. These things dehydrate over time, and they create like a very extreme atmosphere inside of the aerosol. And most people think that, well, then the viruses, they should basically die. They wouldn't be airborne transmissible. But we do know that some viruses are airborne transmissible, and really trying to understand how that is the case. And again, sort of the molecular mechanisms underlying that are really, it's interesting. And so, there's all sorts of questions about how does a virus exist in a droplet that's the same order of magnitude and size, and, not without being affected by the air-water interface, which we know can destroy molecular structure? Or how do things like changes in pH or other composition changes actually affect the structural dynamics of the virus, or more importantly, its viability? So now we can really begin to use what I'm calling in situ computational microscopy to really understand that. Okay, and so with that, I will hopefully I didn't go on too long. I think I might be okay on time. So this is just, I have actually two sets of acknowledgement slides, because there were so many people to thank, and I'm sure that I'm still leaving people out because there were so many people involved in so many different elements of this project. But I had just, I was just so impressed by the response of scientists around the world, and especially the folks in my group. I'll just say it, when we all went into lockdown, and everything was just so, there was such a sense of upheaval of our normal life, and this team just, I don't know if it was coping mechanism or just a really desire to try to make a difference. We all moved into our homes and just became incredibly productive and dedicated to, to trying to do something and learn something about this virus. And so all these just tremendous students and postdocs in my group who were just awesome, especially Lorenzo Castellino, Ziad Gayeb, Amelia, Abigail Domer, Bryn, Mia, Fiona, Tara, Shirley, so many of them. The group of Jason McClellan, who was really just a fantastic collaborator, and scientist, who's really just phenomenal, in his team. Arvind Ramanathan and folks doing the artificial intelligence, and just, again, being, wonderful, willing partners and trying to build new tools to help us explore and expand our understanding of the virus. Elisa Fada and her group, various funding agencies, and of course the compute facilities also. Okay, and then this is just another view of people who I've probably forgotten to thank. As this is Dan Stanzione at the Texas Advanced Compute Center. And with that, I'll close. Hopefully I'm okay on time.
A whole series on what you've spoken about. So it's well worth the effort. And I think we still have some time for a Q&A. Let's start with a kind of technical question from
You bet.
James Van Artsdalem. He says, at the start of the lecture, you showed an equation that looked like it had second and sixth order differentials. Could you explain more about how you developed the equation / model and its significance?
You bet. Okay, so yeah, the equation I was showing was the potential function that we use to describe, again, sort of the interactions of the atoms in the system. So each of those, our atoms, as I mentioned, we're treating them as hard spheres, and they're connected by bonds, which we basically model with like a spring constant. And then two of those, every two atoms that are connected have a bond, and that's described, and every three atoms that are connected form an angle, and that's also described with the spring constant. And four atoms are connected have what we call a dihedral, and that has a torsional term, which is a cosine type thing. And then we have longer range forces, or we have non-bonded forces. So our non-bonded forces are long range electrostatics, which we model with a very simple Coulombic potential. And then we also have the van der Waals sort of collision, which prevents the atoms from, collapsing on top of each other. And so this is this set of sort of ordinary differential equations is then integrated over time. So this is not a method that I developed. This is actually has roots from many years ago, and sort of like molecular mechanics, in trying to use this sort of relatively straightforward representation to understand. To approximate what the atoms in these biological systems, proteins, how they would actually move. And so you have that equation, but then you also have part of that equation are these, it relates to something that we call a force field. So each of the atoms in our system have properties that are different, and how they're built up into residues. So many atoms making different kinds of residues. So we have these force fields that describe in a fairly general way the sort of the molecular ingredients that proteins and nucleic acids, I didn't talk about nucleic acids in this talk, but these are sort of, there are these general categories of biological materials, that people have been studying for, from now many decades. And so what I talked about, so this is now a rather large field of like many, thousands of scientists who are not only using them, but also continuing to sort of, to develop improvements to the force fields. And which is important. So, in terms of like the bigger picture, it helps us, it can help these types of models can also help us do things like design better drugs, for example, or new drugs that might target different states of proteins as they, as they go through their functional cycles. Yeah, so that's
probably a little too technical to describe exactly where the equations came from, because there are actually a lot more equations than you showed, but I think it just,
yeah, I mean, well,
yeah.
yeah, it's actually yeah, because it's sort of like a nested, I guess it's in some sense a little, there's more to it, but it's actually fairly straightforward. I mean, it's a very general representation, and then we use that to compute the forces on the atoms, as they are moving around in time, and that's essentially it. So it's actually fairly straightforward of a method, and it's in some ways remarkable that I think it's in a lot of ways it's remarkable that something that's really relatively so simple can also be so predictive, but that's in part because people have been working to refine these things called force fields, which, have been sort of calibrated against experiment to recapitulate these properties that they can actually experimentally measure.
So he also has some couple of other questions, and one of which is he wants to know, you mentioned how accommodating the Texas Supercomputer Center was in supporting your work. wants to know, where does the funding come from to allow this? And as a matter of public policy, what should be done to ensure that the US continues to have this capability? And also, what did you observe in this respect for similar research taking place in China? So,
Okay.
question.
Okay, a lot of questions. So, all right. So the funding for these, machines generally has come. So these machines, the supercomputers are generally built. They've generally in the past been built by the National Science Foundation, which invests in machines that they build that are open to all academics in the US. And then there's other machines that are built by the Department of Energy and the Department of Defense, which some people can get access to in a more limited way. Some civilians can get access to in a more limited way. But that really provide these, important capabilities in terms of our cyber infrastructure. And so, in terms of investing, I would say, yes, that is something. So under the Obama administration, he came out with, their executive office came out with a commitment to fund the next exascale machines. And that was really important. And then also, more recently, there's been a lot of investment also with artificial intelligence-based methods and machine learning. And so, it's always important that scientists can show, the sort of the real value in continuing to invest. I would say, I feel like, especially because of artificial intelligence really coming into the vernacular and really coming into potentially really great use across a variety of disciplines, that the investments have been substantial. And it's up to folks like me and others to really sort of show that we can, I think, make incredible useful gains out of that and continue to to invest in these next machines. So there has already been a commitment to build the next exascale machine that's going to be, I believe that was an agreement with the Texas Advanced Compute Center. So they're getting sort of the successor to Frontera, which is a huge award. And actually the billion particle respiratory aerosol, I think I can say this is going to be, I hope, one of the driving projects as they build that architecture. So they're keeping that in mind. In terms of other countries, so Japan also, you asked about China specifically, but I'll say Japan has a really fantastic machine at Riken that I believe actually maybe in 2020 actually took over, I think, one of the top fastest machines in the world. And of course, they have a great history in supercomputing and in pushing computational cyber infrastructure, also because they do a lot of weather modeling there. So these machines are useful across, and also with particle physicists and so forth. So with China, I know that they, of course, are also building big machines. And I think actually just in the past couple of weeks that they announced that they now have the fastest machine in the world. I don't remember the number, but it might be exascale. It might be the first sustained exaflop compute in the world. But I think we're going to hear, I've been so knee deep in coronavirus research that I don't know the exact numbers of their machine, but yeah, so they also are in this race for sure. And I would say it is important, and it provides, I think investment in this space is really key, because it does provide potentially very important scientific capabilities, and also links to defense and modeling in a number of different ways. I hope that answered your question.
Yeah, I think it does. I guess the side question is, and probably the answer is no, because the fields are very different, but you may know that yesterday the decadal survey for astronomy was released, and they have a very serious 10-year outlook where they define the priorities in the astronomy community. And And the government's been pretty good about funding things according to that priority list. Is there anything similar in the world of computing, or is it just so, there's so much going on in the private sector and in so many different areas that you can't really have a big master plan for everything?
Well, no, I will say, I mean, these machines are so big and so expensive to build that they sort of,
Have a road.
yeah, they do have roadmaps, and they talk to scientists, like me and others in other fields, about what their requirements are. And you're always trying to project out in terms of like, where are we going and where are we going to be 5 years from now, right? So for what I showed you, that was capability setting, but 5 years from now, that will be more routine, and sort of, and so on and so forth. So in terms of like the scale of the problem and sort of the reach of the problem, so I would say, and we also use, we share these supercomputers, we share with other disciplines also, with people in astronomy, and so forth. So there is quite a bit of coordination. And I think, there's also a little bit of difficulty there because different models will have different codes and different numerical requirements, and they'll map better to different types of compute architectures. Some will need very fast interconnects that the processors can talk to each other in real time while the simulation is happening. That's the case for our type of systems at the big scale. Others will be, they're able to, divide the problem a little bit differently, and they don't need that. So there's these different requirements, and they do really very carefully collect the information and then, bring it back and try to create these machines that really can support the whole sort of long tail of science, And so, yeah, I think it's, the whole cyber infrastructure development in the United States and the world, it really is like an ecosystem, and we try to maximize scientific capabilities, like with many different types of machines, essentially. But it's pretty well, I think it's pretty well planned at the very high levels.
I have a question from Carl Merrill. Carl, you want to unmute your mic and ask your question?
Thanks. That was a heroic, spectacular presentation.
Thanks.
And I worked in this field very early on when people were struggling to look at molecules with X-ray crystallography. And in fact, I remember getting the data from Max Perutz on the effect of oxygen on hemoglobin.
Yeah.
Of us trying to build a model based on that. So it's come a long way since then. The question I have, one of the questions is, in those pictures and illustrations you show, how much of that movement is intrinsic in the wiggling of the molecules just on their own basis, and how much of it's based on extrinsic factors like the solvent molecules colliding with them, which results in what we call Brownian motion? And I couldn't tell whether you could dissect out one or the other, or was they all done in the presence of the solvent? And so that's one question I want to ask, and I have one other question after.
Okay, so that's a great question. So everything that I showed you what was explicitly solvated. So all the atoms, are there, type motion and modeling, where it's a lot of sort of, sampling the different really internal degrees of freedom and working all of that out, like while it's, colliding with the solvent. And then potentially also, other molecules, in the solution, but not really something so long. So, Brownian dynamics is something else. We also do that in my group, but I didn't show you any of that. So everything I showed you had solvent there. And that's one of the reasons why we're more limited in terms of the timescales that we can sample, because we have to carry that around, really sort of slows it down, and it's sort of, you have this more viscous environment essentially. And those rearrangements take a long time.
And another question is it known how many spike proteins have to bind to ACE receptors for the virus to get into the cell to release its nucleic acid? Is it one or is it many?
We don't know. We don't know. So that's one of the things that we're trying to help elucidate with this level of modeling. We don't yet really know the answer to that question. But, you know, if you look at sort of like the curvature of the virion and like the host cell, you can kind of approximate. It's probably going to be more than one connection. It probably might need 3 or 4, but we don't know that definitively. And also, you know, I don't know if you noticed, but when I showed the tomography data, which showed like the virion with the spikes coming out of it, it's not so densely crowded. The spikes are sort of, they can be quite sparse, and they're definitely not regularly placed on the virus. You know, they're sort of, it's highly pleomorphic. So it's got, you know, it can have different distributions, and I would say it's relatively sparse. So the possibility is there that it might only take one connection, but we don't really know that. And I would think if you look at across the average of all particles, given what we know, it's probably more like 2 or 3, I would guess, but we don't actually know that definitively.
And finally, the work you're doing on aerosols is absolutely critical. It's surprising to me that people didn't realize this earlier. There's a whole field of biowarfare where people have been making aerosols, which got us in a lot of trouble with anthrax a few years back. And I just wondered if there's some crosstalk and sharing, since they did all that work, and in light of that, whether there's comparison with other viruses, because I know probably one of the most infectious viruses is measles. And so
yes,
that this differ from COVID-19, and could they approach each other? So that's it. Thank you very much for the presentation.
Oh, thanks for those questions and those comments. And, you know, I would say, I mean, so, you know, people have been studying airborne viruses using, you know, the tools and experimental tools, but as far as I know, these are you all actually, because I actually haven't really talked about this yet. We're about to disclose this. The respiratory aerosol simulations actually are part of another run at the Gordon Bell this year. And we just found out like 2 days ago that we were selected as finalists. And so not next week, but the following week will be when we, you know, really sort of broadly, well, I guess here I'm disclosing it already, but this is like the first time that people have, that we'll be sharing this with people, and that these types of models have actually become possible. And I mean, that's why I think it's so exciting. Yes, we can look at measles, we can look at Kawasaki's disease, we can look at how it's different against influenza, which is more droplet transmission, you know, really trying to understand what makes viruses more airborne transmissible, you know, and so there's so much amazing science there in terms of like the basic science, but then also, you know, the extension to being able to make, again, more evidence-based decisions on whether or not people need to wear masks or under what conditions, and so forth. So yeah, there's so much to be done. And I haven't interacted, just speaking honestly, I haven't interacted with too much with folks in the Department of Defense yet. I don't have deep knowledge on the biowarfare stuff, I gotta say.
Well, we have time for only 2 or 3 more questions. So let's get Frederica Durama's question. She used to be the head of the Air Force Research Laboratory. Frederica, you want to unmute your mic and ask your question?
Hi, Romy. Thank you for the event. Hi, we may have met actually. I visited the UCSD a few times, and I know a bunch of people there from the Supercomputing Center. And fantastic presentation. And also, I wanted to say, since you were at Illinois, I suspect you were in Klaus Schulten's group.
Yeah.
And Klaus must be very, if he lived, he would have been elated to see, because he's one of the fathers that used, in a sense, molecular dynamics. And actually, I interacted with him. He sent, I was at the IBM, TJ Watson. He sent 2 of his students in a supercomputing kind of work that we were doing. So he will be very happy to see all the work that many people, and you especially, are doing. So my question was, are you going to be in the one of the Gordon Bell awardees at supercomputing, and I thought you were going to demur responding to me. So congratulations. I'll be at supercomputing, so maybe you will be there.
That's great.
So,
I haven't decided if I'm going to go because, you know, they literally told us 36 hours ago, and now I'm trying to just sort of shuffle around my schedule, but they haven't even told us when our talk's going to be. So I'm just trying to figure out.
Thursdays, they announce and they're on Thursdays, although they may have shifted the schedule around.
I think it's Thursday. No, I think you're right. I thought they also had some talks on Tuesday, but I think Thursday is definitely when they announce it.
Right. Well, you should come. There are many, many talks, and there will be many people there. Some of the people are where their names were in your programs.
Of course, the slides and
the papers, and I assume in your team. So, well, if you do, we can meet there. But also, I will visit UCSD sometime.
Oh yes,
I know the people at Supercomputing. Anyway,
that would be great.
So a great presentation, and if we meet, I will ask many more questions in person.
Oh, wonderful. Well, thank you for the comments too about Klaus. You know, he was such an amazing mentor, and you can see him in this work. Yeah.
I mean, yes, absolutely.
Yeah,
absolutely. He actually was the one that did molecular dynamics in a medium, you know, related to the questions of the previous questions that were asked.
Mm-hmm.
Okay, we're kind of running out of time, so let me get in 2 more questions. So PSW member Joel Wilson asks, the interaction of aerosol science virology, do you think can provide tools for clinicians such as pulmonary specialists, and perhaps have or will your modeling inform their work or been informed by it?
I would say, you know, yes, I would love to get to that point. And I think we are definitely on our way there, you know, really being, you know, you know, the short answer is yes. The long answer is it's going to take us a little bit to really make those connections very concretely. But, you know, this does surely open up a lot of possibilities. And also, you know, as our abilities to model more realistic systems, whether it's related to SARS-CoV-2 or to the like you're talking about pulmonary specialists, so things like cystic fibrosis and trying to understand what's happening with gelation, for example, in these mucins, you know, a lot of that could be controlled, looks to be controlled by, you know, at the end of the day, you do need to understand where the atoms are. So it could be like with cystic fibrosis, that it's something with calcium regulation. And that's because calcium, you know, it interacts differently with the side chains of the mucins, you know, at a very detailed level. And that changes, it causes it to form a gel, and it changes completely then the physical properties of that system and affects the ability, you know, of the lungs to operate and so forth. So, you know, our work connects to the work, I think, of pulmonary specialists and so forth. And I foresee a future, you know, where we have computing really sort of at the patient bedside with physicians in an informed way. Machine learning will help that because it will be able to hopefully make some of our calculations happen faster. And in a more rapid way, that the turnaround could be sort of more in real time for, you know, for various sort of, I guess, sort of more on precision medicine type things. But yeah, so I think we will get there.
So I'm going to close with one last question from SW member and lecture sponsor, Tim Thomas. I think it's a good question to close with. So what signal might the flip? Oh, sorry. Go ahead, Tim.
Yes. What causes now my brain's going to slip me, the receptor domain to slip out? Does it do so in the presence of the human cell with an ACE receptor, or does it do it in the aerosol, or does it flip around from one to the other state, down and up?
So it's a great question. You might need to mute. Yeah, so that's one of the things we're trying to understand. So it appears that, it does have the ability, I think, to spontaneously, open to different degrees. At least one every once in a while can pop up. Also 2 of those can come up, and actually even 3 of the receptor binding domains. So one of the things that we think is happening, but we haven't published on this yet, but that we're trying to study is that, and we've done so much, this part I couldn't even include. So there are these things coming off of the host cell called glycosaminoglycans. So these hosts, the human cells, not only do they have ACE2 there, but they have these sort of long sugar chains, this glycocalyx, this like oligosaccharide sort of layering. And we think, and others, there's some evidence that suggests that as the virus gets and the spike protein gets closer to the cell surface, that there are these that these glycosaminoglycans actually can interact with the spike, and sort of, I would say, almost like it looks like they almost look like they're tickling at the spike, and that could actually help to promote that receptor, the transition of the receptor binding domain from the down to up conformation. But it does appear that it's very likely. It's a good question. So I think that in most cases, that the spike in the wild type spike is actually closed, and it's something that happens when it gets into particular environments that sort of cause it to open. And I think it's not necessarily the interaction with ACE2, but it might be before it even gets to ACE2, it reaches these other parts of the cell that are coming up, in the environment of ACE2 that sort of help prime the spike to open. And then that happens if it's when that happens, if it's near an ACE2, then it binds and it's stuck and everything kind of goes. So that's something that we're also sort of modeling and trying to understand, but more, once the virus gets actually, to the host cell. And then in the aerosol, we don't know either. And I think that's a really fantastic question. I mean, I would guess that, so we have all these hypotheses in any case that are, that we're trying to flesh out. I think actually, it's in these respiratory aerosols that these mucins are sort of, they're protecting. They appear to be sort of like interacting with the spike and sort of protecting it from damage, but also maybe helping to keep things sort of tightly closed and prevent opening. And that would then, when actually that aerosol particle gets absorbed onto, inside the deep lung, that then it can actually, once it's deposited there, then it can actually sort of like start to move around and actually interact with the ACE2 and the glycans on the host cell surface. Anyway, which is a very long way of saying we don't actually know yet. We just have a lot of hypotheses that we're trying to flesh out in simulation and also together with our experimental collaborators.
Well, let me encourage you to keep trying.
Thank you. Yes, thank you.
We'll expect an answer in a year.
That's right. Yeah, we'll do our best.
Well, thank you, Romy, for a terrific talk. There are so many questions, so we could probably go on for weeks. But
oh, well, that's good.
Get back to the lab and do some work. So,
thank you.
Although we regret you can't be with us in person, you have a rain check to join any PSW event in the future. Would include the pre-lecture reception and dinner, of course, on us. And even if you can't be here to coincide with a PSW event, if you let us know, we can arrange something and enjoy dinner with you at the Cosmos Club and PSW members. With that, thank you and let you go. Maybe I'll see you later. But take care. Everyone else, before you go, we have a few important notes of the upcoming lectures. The fall lecture series has been posted to the PSW website. The 2,449th meeting will be in 2 weeks on Friday, November 19th. The speaker will be Robert Beyer, Keenan Professor of Applied Physics and Photon Science at Stanford University. He will be speaking about laser-driven particle accelerators fabricated on a silicon chip. This meeting will be held in person at the Cosmos Club, as noted in my opening remarks this evening. The meeting will adhere to COVID-related rules of the District of Columbia and the Cosmos Club. Proof of vaccination will be required to attend, and masks will have to be worn throughout the lecture. Details will be posted on the PSW website and also will be emailed to the membership. Thereafter, we will hold meetings in person when possible, subject to the ability and willingness of our speakers to travel, the DC and club rules, and several other factors. Information will be posted to the PSW website and PSW social media on a meeting-by-meeting basis, and also will be emailed to the membership. The 2,450th meeting will be on Friday, December 3rd. The meeting will coincide with PSO's DuPont Summit, to which all PSW members are invited without charge. The speaker will be Francisco Cordova, director of the Arecibo Observatory. He will be speaking about the observatory's history and its prospects for the future. The 2,451st meeting will close the fall lecture series on Friday, December 17th. The speaker will be Sean Andrews, astrophysicist at the Harvard-Smithsonian Center for Astrophysics. He will be speaking on the current understandings of the processes of planet formation, both around our sun and around other stars. Join us for these meetings. And finally, before you go, let's thank tonight's crew, Connor, Ann, and Robin Taylor for putting on tonight's event. And with that, I will now adjourn the 2,448th meeting of the Society. I wish everyone a good evening, a good morning, a good afternoon, or a good night, wherever you may be. The meeting is adjourned.